నేనైతే నేను క్లాస్లో ఉన్నాను చదివిన తర్వాత మాట్లాడేది ఏమీ లేదు. రెండే సంతకం పెట్టడం, రిజిగ్నేషన్ లెటర్ పెట్టడం, రెండే అని చెప్పారు. మాట్లాడేది ఏమీ లేదని చెప్పేసి అంతే. రెండే నా దగ్గర మాట్లాడేది ఏమీ లేదు. మాట్లాడేది ఏదంటే సోమరాజు గారితో దీని గారితో మాట్లాడుతున్నాను. నేను ఇంక మాట్లాడకూడదు, మధ్యలో ఉన్నాను నేను క్లాస్. Hello, dear students. Good morning to you all. Today, we will try to conclude the molecular biology classes. The last one and the most important one to understand about a very, very important topic, that is polymerase chain reaction. Polymerase chain reaction. This is popularly called as PCR test. Even outside population, they say PCR test, big test, and which costs about 4,000-5,000 rupees cost. These tests, they are highly sensitive tests, highly precise tests. Definitely, they conclude whether a person is having a particular disease or how much the quantity of the viral load and etc. The bacteria loads and other things, and a very useful information will be given by this particular test for the sake of treatment by the clinician, and he gets a lot of information about the patient. To know this particular one, the initial one is, in the previous class we have discussed that recombinant DNA. Recombinant DNA. The recombinant DNA, in the recombinant DNA, we have understood that two different kinds of DNA molecules are joined together, and we try to express it in the intact cells. Let them, it may be a bacteria or it may be a human cells, cultured cells. This is recombinant. Parallel to this, to synthesize proteins, there is another technology called as monoclonal antibody technology. Monoclonal antibody technology. In this technology also, some material, some needed antigen can be introduced into some growing cells, continuously growing cell lines, they say. Into those cultured cells, they introduce a particular antigen, and against that antigen, antibodies can be continuously be produced, and that is another technique called monoclonal antibody technology. And these two are very, very important technologies in the raising or synthesizing proteins for either diagnostic purposes or treatment purposes. So this is about having the fundamentals on this particular science. We shall go for the next applied science. This is what is called PCR, polymerase chain reaction. Here there are two words you should try to understand. One is polymerase and another is chain reaction. Chain reaction. Chain reaction we also heard in the nuclear bombs technology. One molecule strikes two molecules, two molecules strikes four molecules, four is going to be striking 8 molecules, like that exponentially. So this exponential growth is also being utilized, is being present in this particular technique. And then, as I said, one is chain reaction, exponential growth. And what is that chain reaction? Polymerase, with the help of polymerase. What are the polymerases we know in our molecular biology? There are mostly important ones are 3. DNA-dependent DNA polymerases. DNA polymerases. DNA-dependent DNA polymerases. What for these are mainly, what is their main function? One DNA molecule of a parent cell can be completely synthesized into another DNA molecule, which can be utilized as the central nucleus for another dividing cell. In the cell division, to completely produce a nucleus, a replicate molecule of a nucleus, we need DNA polymerase. We thoroughly understood them about them. In the prokaryotes, how they are, and in the eukaryotes, how they are. In the eukaryotes, we also learned there are about different kinds of DNA polymerases, 1, 2, 3, and 4 and 5 also have. And then this is about DNA to DNA production. This is also called as replication. In the replication, you know one polymerase, that is DNA polymerase. DNA polymerase. From DNA, and we learned what is called as central dogma. Central dogma. In the central dogma, we understood DNA containing many genes. And these genes are all having the signal in the form of polynucleotide sequences. And these sequences are transcribed onto another movable polynucleotide, and we have named it RNA, where there is a small difference when compared to that of the DNA, that is the presence of uracil acid and abnormal or rather uncommon nucleotides presence, and usually this is a single stranded. So for the synthesis of an mRNA or tRNA or whatever the other RNA, that is the rRNA, any one of these 3 types of RNAs, we need another polymerase. That is joining one nucleotide to another nucleotide. By always the bonding is, we have been learning phosphodiester linkage. So for this purposes, that particular polymerase is, now we call it as RNA polymerase. RNA polymerase. So this is another kind totally. And there are also differences, prokaryotes and eukaryotic. RNA polymerase is a small difference. And how they transcribe is also there is a differences. We learned thoroughly. Then there is now another kind of polymerase we have learned. Taking instructions from RNA, you can get a DNA strand. This is usually a template strand. But in our central dogma, for a long time, we are under that impression that the message can never go back, but it is going back only after learning about the various viruses. And one great virus which belongs to the same category is our now COVID-19 as well. And they do have, and HIV is another one. And these deadly diseases, they have an enzyme called as reverse transcriptase. Reverse transcription. That is, from the mRNA, you can get back the gene or otherwise the template strand of the DNA. And this process is also brought about by a particular specific polymerase molecules which are present along with these viral particles. So this is what is called as reverse transcriptase. But this model we have been using for our purpose, and it has given a great fruits in our medicine. How from the mRNAs, from the proteins which are lacking, we are trying to decipher the mRNA. We are able to synthesize the mRNA. And from the mRNA, we are able to now, with the help of reverse transcriptase, we are trying to produce a DNA strand. If we have one DNA strand, with the help of DNA polymerase, we can produce a second DNA strand. So now the double helical DNA molecule we are able to produce. And this piece of double helical molecule of DNA of our interest producing a specific protein or specific material that is needed for our treatment or diagnosis kits preparation. Now we can introduce, and that is what the recombinant way, and then we can produce a large quantity, no doubt about it. But while directly to know about various the viruses, whether they are present in the body or not, and directly we cannot test it. The material in the form of viral in the blood, they are present, is so few copies of RNA molecules. very small copies of RNA molecules. Unless few millions are there, we just cannot have a nanogram of material. And with great precise techniques, only we can identify that material. So to identify this material initially or for some other purposes or treatmental purposes, we need to, or otherwise, to identify, we know the fruits out of this technology a little later. So now, what is the basis behind it is, suppose we have few copies of either RNA or DNA, only few copies, which cannot be identified by the general techniques. If it is magnified, the more number of, from few 10 to 20 copies of DNA and RNA, if we can produce few millions copies of RNA or DNA, then we are able to identify that particular material by conventional methods, our methods, our specified methods. So now, these methods, now in this idea, initially, Carey Mullis, the scientist is Carey Mullis, in 1989, he has discovered this polymerase chain reaction. Polymerase chain reaction. And without any hesitation, the Nobel Foundation has given him a Nobel Prize in 93. When he has invented in 89, within short time, 93, they have given the Nobel Prize. So this is, what is it? Come on. In vitro DNA amplification procedure. PCR is, note down, in vitro. In vitro means outside. In vivo is inside the animal being or otherwise organism. So in vitro DNA amplification procedure. Millions of copies of a particular sequence of DNA, that is a gene of DNA, or a small one point to another point, a piece of DNA can be identified, can be multiplied. So this process of amplification from few copies of RNA or DNA, millions of copies to produce what we use, the technique is PCR technique. Polymerase chain reaction. Polymerase chain reaction technique. This is otherwise, it's a Xerox machine for gene copying. More and more number of genes can, that is copies of genes can be synthesized. But for this purpose, what we should know is, for that gene, this side and that side, the flanking sequences, they say. Flanking sequences of the gene of interest, the gene whatever that we want, that particular areas should be initially, we should know about it. And these others, that side and its side flanking sequences, the primary sequence must be known to us. Once it is known, that has to be synthesized in the laboratory. And this is what is called as primers. On either side, we need for this technique two different primers on either side of the gene of, or the polynucleotide chain of interest. So this is one basic criteria we should have before we go for the polymerase chain reaction. And what are the steps in, now we'll see. Suppose we know this DNA, a piece of DNA, double helical DNA. In this, the main idea I will show you. So initially, any polymerase to act on this DNA molecule, what is needed? Both the strands should be unwound. For this purpose, in the animal being, we have so many single strand binding proteins, helicases, topoisomerases, all these things forcibly widens these two strands apart from the bound hydrogen, intra-hydrogen bonds are being cut, which are being associated in the two nucleotides. నేను క్లాస్లో ఉన్నాను, తర్వాత నేను మాట్లాడతాను. చూసారా ముందు So now, with this, the two strands should be separated out. Both the strands should be separated. And once both of them are separated, then the polymerase can act on. And from the within the vicinity of the cell, the nucleotide phosphates are being brought, energized nucleotides are being brought, and this polymerase, initially what is needed is a primers are needed. That's why just now we have discussed it. The extreme sequences should be known, and to their sequences, the base pairing polynucleotide should be synthesized, and that is what is called as primer. I said, so the primers need to be present in this. So in this place, not only widening these two, and then primers are needed, the enzyme is needed, that is the polymerase enzyme is needed, and the nucleotides are needed. So now, normally in the cell, this unwinding process is done by the enzymes, other enzymes like helicases and topoisomerases. But here, it has to be deliberately done. How it can be done? It can be separated by, we know, physical technique. We learned also the DNA melting. That is, the DNA molecules are widely separated by subjecting them to the heat. So at a particular different species of DNA have different melting points. This statement we have learned in the first, second classes. So now, once we subject it to the heat, then they at a particular point, say about 90 degrees or 94 degrees, different species are having the capability to widen them out, or otherwise separate them out. And then these exactly base paired, the primers will try to come nearer to those exactly aligning or otherwise exactly base paired region. They come nearer to that place, and they can search it, and they can stand before it. But the bonding cannot happen initially. And once they have come over there, this is what is called as the second process is annealing process, and then annealing process, and the primers are all going to come and stand at their position. And then the next one is the enzyme will come. But at 94 degrees, the normal cellular polymerases cannot do polymerization process. Normal polymerization process needs, in our human being, we need the temperature nearer to the 37 degrees temperature. But suppose by any chance, can it be possible that 90 degrees temperature can occur inside the body? It is not possible. So how the polymerization? The invention of one special polymerase. There are, to our wonder, there are what are called as hot springs in the nature. In the hot springs, their temperature is nearer to the 100 degrees. Even in that hot springs, we are surprised to see certain organisms are living in it. Normally, our tissue, if it is put in boiling water, it is going to completely destroyed or denatured or completely dead, completely dead. And then this, there are a few organisms which are surviving in the hot water. So now, in these hot water surviving species, one such species is Cerumus aquaticus. And this Cerumus aquaticus species is containing also polymerase. And that particular polymerase is able to polymerize this particular complete strand. So the next step, you see, there is an extension. And this Xeromus polymerase aquaticus enzyme coming over here, and one by one following the base pair rule, and one by one it is linking one nucleotide with another nucleotide. The nucleotides are present in this reaction mixture or in this in that chamber, particular chamber. So we deliberately add excess of nucleotides into this reaction chamber. So now this is about the DNA polymerase of Xeromus aquaticus. So continuously this go on producing, and then from initial 2 strands, after these 3 steps are finished, we are getting two different DNA molecules. So then again you subject them. Suppose you cool it, then what will happen is they will anneal. They wind one to another, and again DNA molecule, 2 DNA molecules, you can have it in the first step level. So now after this process, from 1 DNA molecule you got 2 DNA molecules. Now these 2 DNA molecules, now you subject it to again denaturation. Then they will be separated into 2 strands, and then you make the annealing process, and then enzyme polymerization and extension process, and you will get now 4. So in the second cycle, this is cycle 1 by 1 cycling. They are say this is 1 cycle. 1 cycle is completed, you will get 2. If on this you do the whole process, if second cycle is completed, you will get 4. And the third cycle is complete, you will get 8. And by about 20 to 25 cycles are complete, you will get million copies. And these million copies you can see, and then to identify them, you have techniques to identify DNA material. The material, DNA material, can be identified by electrophoresis technique. Electrophoresis technique, coupling with what is called blotting technique. And this type of blotting, electrophoresis and blotting, and identifying the radioisotopic materials, whether that particular DNA is present, the sequences are present, and this technique called as a Western blot, northern blot. For DNA, it is northern blot. For DNA, it is northern blot. Southern blot, then Western blot, and for mRNA it is northern blot. For DNA, southern blot. For proteins, it is Western blot, and for the RNAs, it is the northern blot. So these are the blotting techniques. With these coupling these blotting techniques, you can identify them. A particular specific sequence, or a specific sequence of a particular, say, a virus, or for example, tuberculosis. A tuberculosis is having a few genes, and such genes, whether they are present or not, initially targeting to that particular genes, we will multiply the DNA material present in that given sample. And once the DNA material is multiplied, whether that TB specific sequences are there or not, we will finally identify it. And that is how a TB, in very few number of TB present, in normal tests we cannot identify, but in PCR we can identify. So at the earliest stage of infection by TB or viruses like HIV, or like HPV, or like even COVID, now we are using this PCR test. So what is the test we are using for COVID now? The gold test is PCR test. And in this PCR test, COVID is having the RNA, and hence initially this DNA can be multiplied. So RNA has to be converted into DNA. How can we convert? Now reverse transcriptase enzyme is needed. So here this PCR is RT-PCR, reverse transcriptase based PCR. So for COVID and HIV, it is RT-PCR. Again, for HIV and HPV, COVID, or HCV. HCV is a hepatitis C virus. It is a jaundice virus. HIV, you know, immunodeficiency virus, AIDS virus, and COVID, you know, this particular coronavirus. It is producing the COVID infection. And these viruses are having RNA as their genetic material, and hence RNA is converted back into the DNA first. And that for that purpose, you need reverse transcriptase. And hence, reverse transcriptase is also present in this test. And now, this is called as RT-PCR. Initially, we should know about PCR. Fundamental is PCR. And for this PCR, various modifications can be done like this. And then, for diagnosis purposes, the most important test now all over the world being used is this polymerase chain reaction. So, the steps are: step 1, denaturation. Each procedure will have a small changes, 1 minute of 94 degrees heating, and then slowly it can be reduced back. And there present the primers in that solution. The primers nearly comes and adhere to the exact point. And from at this level, thymus aquaticus starts functioning in the extension. And then for 2 minutes, again, at a bit of 72 degrees temperature, this polymerase slowly adds up, polymerizes the nucleotides, and it is continuous, and the whole process completes from either side. And then you get two copies of DNA molecules. Again, you will subject it to 94 degrees temperature. So cyclically, you alter it, and specific timings are there. And already here the mixtures, the targeted gene flanking sequences, primers. This is the most essential one. Then it can go, and on either side of that particular gene, they can adhere, they can add on, they can join, and from there, basing on this, the polymerase starts working out, and all this region, this is taking as a template. This is a coding strand will be completed. The second anti-template coding strand will be completely synthesized, and two such molecules occurs. Two such molecules will be produced. And again, this whole process, again, they widen together, primers will come, join, and then extension occurs, and the 4 molecules. So this is the PCR amplification. And this is what is called as exponential amplification. From 1 copy, there will be 2, and in the second cycle, there will be 4, in the third cycle, 8 to 16 to 32 and 64, etc. And for the 35th cycle, you see, 35th cycle, almost 68 billion copies are being produced. So we just cannot imagine. So if this is so, it is a long process. It may take a few hours to few more, some 6 to even 12 hours or even more number of hours, because this process continues. So 30 cycles, 1 billion copies, 1 billion copies of DNA molecules can be identified. Now, what are the applications of PCR? Mutation screening. Suppose a gene of a particular individual, whether any mutation is there, to identify it, you will use it, the PCR. The drug discovery, new drugs discovery, for that purpose, you need PCR. And then, for the classification of organisms, also, we are using the PCR. Genotyping, genes typing purpose, you need the PCR. Now, later on, the molecular archaeologists, that is, suppose we have some fossils, and what is the age of this fossil, and when it would have been, and is it really an organism of old time, etc. To know about it, our PCR is being used. And molecular epidemiology, to screen, to identify a virus here in the India, and a virus here in the US, and a virus in the South America, rather the Russia. all of them are same primary conformations or not, or else from one place to another place, from which place to which place this would have been migrated. Do you know about that? Science, you are using the molecular epidemiology, the PCR technique. And then molecular ecology, the ecosystem to match various the organisms in the various areas. That is also a PCR. And in the bioinformatics, PCR, genomic cloning and site-directed mutagenesis. Gene expression, we have studied in the previous class, that is gene expression studies. These are all basic fundamental researches. Now, our medical applications. Genetic matching. A particular individual claims, if this man is my father, and people say, probably you must have been heard. We are taking the sample of both of the blood, and we are trying to match DNA finger, DNA matching techniques. That is genetic matching. Genetic matching techniques. And the detection of pathogen here, where we are very much interested. Various tests, mainly tuberculosis and HIV. These both infection diseases are being identified very, very, very early. And when various previous gold methods are not able to be identified, this very precisely, specifically, sensitively, specifically is being identified. And for that purpose, PCR became our great weapon in the medicine. And prenatal diagnosis, yes, this is genetic diseases. Genetic diseases. For example, a sickle cell disease, or anencephaly. And these ones, if they are there in the mother's womb, then early, suppose. But if the fetus is grown beyond 24 weeks of pregnancy, then the termination, that is aborting it, will cause damage to the mother. So such a abortion we cannot afford to do. So before that, if we know that fetus or that baby pregnant, that one, newly growing one, if is having a disease, if is known to us, then we can conduct an abortion, and probably it will be safe for the coming up child as far as the family which is supposed to grow such a crippled child. And this is what is prenatal diagnosis. How can we do this prenatal diagnosis? By taking 1 or 2 cells of villi samples, or amniotic fluid cells, few of them, 4 or 5 cells, if we can harvest them by taking a small quantity of amniotic fluid, and from these few cells, if we can multiply with the help of PCR a lot of DNA, and then with the help of probes for the diseases, if we can identify whether that diseased gene is there or not, we are going to identify inborn inherited diseases, presence of inherited diseases. If it is certain, then we can abort it. Then another one is DNA fingerprinting, matching of the various regions of various chromosomes, and different individuals or even different animals. So this is all called as DNA fingerprinting. Especially these are useful. These are useful for daily our daily, especially the forensic medicine purposes. And the last one is the gene therapy. The most promising one, the most costliest one is the gene therapy, where the diseases, genetic diseases, where we just so far unable to treat, we used to brand it, and we used to say, we are sorry, you have to live with this problem. But for that purpose, the gene therapy is now becoming a popular therapy and a promising therapy, where a recombination with the viruses or some plasmids like that can be, the genes of interest can be and be cloned, and then the genes can be introduced into these particles, larger particles, and they will be installed, they will be placed in the organism where that proteins are deficit, and they live for a certain time. They live for certain time. Till that time, we can have that particular protein. That means that action can be there. Here, we are very much interested. So many scientists are making their effort here to progress, to increase the lifespan of producing the proteins by these small miniature creatures, that is the viruses or others, and continuously to produce for a long time. So this is about gene therapy. And here our now you can note down that for specific nucleotide sequences of bacilli can be identified where in the detection of pathogens. And genetic disorders, we are identifying mutations. So for the purpose of diagnosis, there's diseases like sickle cell disease, beta thalassemia, and cystic fibrosis. Again, sickle cell disease, beta thalassemia, and cystic fibrosis. And another prenatal diagnosis, that is by amniocentesis or villi sample and harvest few cells, and then DNA is there, harvest it, multiply it, and diagnose the disease. And then another one is cancer detection. The material where the growth is there, take it out, and then try to see the mutations of P53 gene and retinoblastoma gene. These two are cancer suppressor genes. Normally, if they are present, they will halt the cancer. But if they get mutated, what will happen? Because of the mutation, their function is lost. So the suppressive capability for cancer is lost by those cells, and means the cancer is progressing. So because of mutation of the normally functioning proteins, there can be, or abnormally functioning, that is oncogenes normally present in the dormant stage. As long as they are proto-oncogenes, rather proto-oncogenes are there. But once they are mutated, they become oncogenes. Proto-oncogene will be there. It is harmless. But once it is mutated, it becomes an oncogene. Once oncogene has been mutated, and then it is trying to produce the continuous continuous multiplication of that particular cell. So this is what is as a clonal clarification, that is progression of these cells, proliferation of cells. That is what is continuous. Without any sense, it is going to produce more and more number of cells, and that is cancer. So the oncogenes. And again, oncosuppressor genes, some others are there, and they also, if mutated, the suppression is lost, and hence the cancers can progress. And then last is the medico-legal cases. DNA from hair follicles or blood spots which are being present in the scene of offense can be taken, and that particular material is matched with, we will identify 2-3 members as the culprits. So they might have done a crime. So then all those 4 members or 5 members, samples, blood samples also will be taken, and in the crime scene, these blood samples which are being present, they will try to match them, and they can identify the culprits, and also we can match with that blood but is the same that of the deceased, that is the dead one or otherwise attacked one, and his blood and the match the scene of offense, blood spots matching, then it is also will be identified. And this is how the PCR is really revolutionized the medical science. So now, as I said, the RT-PCR, reverse transcriptase PCR. This reverse transcriptase PCR can be completely finished, and then the obtained material can be identified that it belongs to TB disease, TB bacilli. It belongs to HIV bacilli. So this is a qualitative PCR. Qualitative PCR. So there are two different PCRs: qualitative PCR and quantitative PCR. Diagnostic techniques. In the diagnostic techniques. So in the there is another word that is being used: real-time RT-PCR or real-time PCR. Both of them, they will also able to identify how many number of bacilli they might have started with. So what is the density of the bacilli in the blood? In the blood, what is the density of the bacilli, or what is the density of the viruses? This is what is called as a real-time PCR. This is real-time PCR. So with this, the PCR is being completed. And then the last is, we know now the whole genome of a human. And genomes, complete genome, this primary sequences of various organisms, and like Drosophila. So then from that, the various experiments are being done on genetic techniques. And then this is the completely unraveling the whole process is called as a genome project. And this human genome project, I also told you, reminding you that the whole human genome project has been taken up under the leadership of Watson, one of the person who invented DNA. And then it is a multinational: Germany, Japan, England, and then France, and Canada. Various centers are there, and they are all trying to work out what are the recent advances in the gene therapy. And once knowing about all, to know about how diseases can be prevented, how diseases can be cured. That is ultimately our aim. So for that purposes, the therapy that can be offered is gene therapy. So advances in the gene therapy, we will wait for. And then this is how the molecular biology has revolutionized all the sciences. If you want to understand the pinpointing the area in the cell, to know about it, even in the anatomy, you need a PCR. To understand the physiological processes, also you need PCR. And biochemistry anyway is integral part of PCR. Even in the pathology and medicine, everywhere, the molecular biology has come in. So in the olden days, the pathology, the diagnosis by histopathology is the final one. Now, histopathology is not final. Now, molecular pathology is the final one. So, basing on this molecular pathology, the therapy and so many monoclonal antibody reagent, rather the therapeutic agents have come out, and they have revolutionized the cancers. And in 90s and 2000s, where the cancer is not able to be cured, at least certain types of cancers are able to be cured with the help of, or otherwise remissions able to be obtained by the using of these antibodies for cancers, which are being produced either by recombinant DNA technology or by a way of monoclonal antibody technology. Both of them are brothers, and they produce large quantities of proteins. So with this, you please go through the whole molecular biology, and molecular biology is a very, very very, very advancing science in the medicine. And without the molecular biology, no science can be explained, especially our medical subjects. No subject can be explained. Every subject now is having molecular biology basis. Unless that, we cannot understand. So even though initially it's a little bit difficult to understand, but once you go through two, 3 times, then you will not forget, and it will be coming into your control. So hope you will be you had understand that this particular topic, and then try to read well again and again. I am reminding you the examinations are coming soon, and the governments or rather the universities want to conduct the examinations in the due time. They don't want to give any postponement or etc. And hence you need to work hard. For a few days or for 1 or 2 months you completely relaxed, but now you need to work so hard to compensate that relaxation. So please do well, read well, and hope you will succeed in the examinations, coming examinations. Thank you very much, and good luck.